I would like to today's session by extending a very warm welcome to our guest speaker, Professor Colin Hill, who is going to talk to us about probiotics and their role in influencing human health. Professor Hill, having spent almost his entire career researching in different areas of microbiology, is a leading authority on probiotics. He is responsible for promoting rigorous research in the field, having published more than 550 peer-reviewed papers and holding 20 patents in this area. Suffice to say that a complete list of Professor Hill's achievements and contributions to the field of microbiology need a whole webinar of its own. Professor Hill, we cannot express how thrilled we are to have with us today. I hope that the webinar not only inspires all of us, especially the young minds in our audience, but also sparks discussion around the extremely important topic of probiotics and their effect on health and wellness. Please allow me to express my gratitude to Professor Geeta Trilok Kumar, the Director of Institute of Home Economics, under whose constant guidance and support this webinar is being held. We are also fortunate to have Professor Arvind Deshmukh, the President, All India Microbiologists Society of India, MSI, and Dr. A. Archana, the President, Delhi Unit of MSI, with us. Professor Deshmukh has very generously taken the time today to be part of our webinar. I would request Professor Deshmukh to kindly share with us a few words. Professor Deshmukh.
Thank you. Thank you, madam. Really, it's a pleasant morning for Dr. Colin and a pleasant afternoon for all of us. The reason is that he is talking on a very, very interesting topic. Because nowadays, people understood the importance of probiotics as well as prebiotics. And during the period of Corona, everyone was talking in India about Ayurvedic medicine and probiotic role in improving the health during Corona. So definitely people are knowing it, but as Dr. Colin's study during his life, depth of the knowledge, he is going to put forward today. And it's a best opportunity to all students from India to understand this interesting field in microbiology, that is a probiotic. Whether we should call it as a medicine or not, that is again the question, but Dr. Colin will answer it. So, I appreciate the efforts taken by Dr. Sunita Agrawal by arranging an interactions at an international level and giving exposure to the young budding scientists of India. I am thankful to Dr. Archana ma'am, who is the President of Microbiologists Society at Delhi. Really, she has kept a very good academic environment in Delhi, so that continuously there are activities from Delhi chapter of Microbiologists Society. At the beginning only, I express my thanks to Dr. Colin for coming over on our platform and for giving consent to deliver lecture on a very interesting topic. Thank you again. Thank you everyone. Thank you attendee also. I can see about more than 95 attendees are here today. So I welcome all those and thanks to all. Thank you.
Thanks, Professor Deshmukh. Dr. Sunita Agrawal, Program Coordinator, Star College Scheme, and State Coordinator, MSI, is also with us today. She has been a source of inspiration, and without her, this webinar would not have been possible. Now, without further ado, I would like to invite Dr. Sunita Agrawal to welcome and introduce Professor Colin Hill. Dr. Sunita Agrawal.
Thanks, Dr. Pratistha. Good afternoon, everyone. It's my privilege and honor to introduce Professor Colin Hill to this august gathering. Professor Colin Hill has a PhD in molecular microbiology from University of Ireland, Cork. Presently, he is the Professor of Microbial Food Safety in the School of Microbiology at University College Cork, Ireland. He has been an SFI Principal Investigator since 2002. He is also a Principal Investigator in APC Microbiome, Ireland. His main interests are in the infectious disease, particularly in defining the mechanism of virulence of foodborne pathogens and in developing strategies to prevent and limit the consequences of microbial infections in the gastrointestinal tract. Professor Hill was awarded a DSc in 2005 by the National University of Ireland in recognition of his contributions to research. In 2009, he was elected to the Royal Irish Academy, the highest honor for an Irish academic. In 2010, he was elected to the American Academy of Microbiology, and together with his colleagues, Professor Gerlach, Professor Paul Ross, and Dr. Catherine Stanton, he was awarded the Metchnikoff Prize in Microbiology. He served as president of International Scientific Association for Prebiotics and Probiotics from 2012 to 2015. He has highly cited publications to his credit. Thanks, Professor Colin, for accepting our invitation and sparing your valuable time to deliver a talk on probiotics, alternative medicine, or an evidence-based alternative. I welcome you once again to the IHE on behalf of our director, faculty, and students of IHE, and President Sir, member Microbiologists Society of India. May I now request Professor Colin Hill to deliver his talk. Sir, please.
Thank you very much. Thank you. It's really an honor for me to speak to this group. We have had a lot of really talented Indian students and postdocs in our group, and I visited India, and I'm really impressed by the work that goes on in your country. So I'm really honored to be here, and thank you again for that very warm welcome. I'll share my screen now. I hope this is working. What I chose to speak about in this meeting are probiotics as an alternative medicine or as an evidence-based alternative, as Professor Deshmukh has just explained. And I live here in Cork, and I work here in Cork in Ireland at an institute called APC Microbiome Ireland. So what I like to do is set probiotics in the context of the microbiome. One of the most important interactions that we have, of course, with bacteria is with the microbiome, and probiotics work on that stage. So I'd like to explain that as well. So probiotics is a very hot topic. This is a search I did just earlier this month on Web of Science, and there were over 30,000 papers have been published on using the term probiotic, and over 3,500 just in 2020 alone. So every year, literally thousands of new papers are appearing. So this makes it a very complex field to stay current with. And even if you limit the search to probiotics and humans, then you still get almost 1,000 papers in a single year on probiotics and humans. But of course, this doesn't even, this pales into insignificance compared to the amount of papers published on the microbiome or the microbiota. Both terms are used. Nearly 120,000 papers have been published on that topic, over 22,000 in 2020 alone. So if you wanted to stay current with the literature in 2020, you would have had to read over 25,000 papers, and obviously nobody is capable of doing that. And so what I'm going to give you is a very biased view of the probiotics and the microbiome, most of it based on our own work here in Cork, but involving some other work as well. So you know, as microbiologists, But most people don't realize that we actually live in a microbial world. Microbes appeared on the planet 3.5 billion years ago. We only came in the last 1 to 2 million years, which means we arrived at a few seconds before midnight on the world clock. But microbes made most of the oxygen that we breathe and have shaped the planet that we live in. And we have evolved into this microbial world. And if you want to understand how important our microbes are to us, all we have to consider is that mother's breast milk contains many oligosaccharides, human milk oligosaccharides, which the baby cannot digest, which are targeted at the microbes in the baby's gut. So one of the first concerns of every human mother is to feed her baby's microbes as well as feed her baby. And that gives a sense of how important the microbes are to us in terms of our evolution. And it's interesting to note as well that we share a language with microbes. We have adopted microbial chemicals as our signaling molecules. So microbes produce serotonin, acetylcholine, GABA, butyric. We didn't invent these molecules. We adapted them from the microbial world. So we speak the same language as our microbes and we can communicate. I wrote an article earlier this year, it was last year now, called You Have the Microbiome You Deserve. And what I was trying to say in this article was every one of us has a unique microbiome which is shaped by events in our lives. Some of them are listed there on the right. You can see whether you were breastfed or formula fed, your age, genetics, birth mode, whether you've been on antibiotics or not. All of these things shape our microbiomes. And now you have a microbiome which is completely unique to you. And of course, any prebiotic or probiotic or dietary component that you ingest has to engage with this microbiome. There's a lot of numbers associated with the microbiome. I won't go through them here, but things that always catch people's attention, I think, is that we have as many microbes in the human body as we have human cells, and as many viruses or bacteriophages as microbes. We've probably 150 times more microbial genes in the human body than human genes. Most of our microbiome is in the GI tract, and as I've already said, it's unique to every individual. But it's quite stable within each individual. So you normally assemble your microbiome in infancy, and big influencing factors are whether you were born by C-section or by standard vaginal delivery, whether you were breast or bottle fed, at what time you weaned, your environment, whether animals are present in your environment. But within a short number of years, you assemble a microbiome that essentially stays with you for life as a kind of a microbial organ within your body. Now, this can be affected by diet and antibiotics and disease and environment, just as any organ in the body can be affected. And when we age, when we get older, the microbiome tends to dissipate or collapse a little bit, and we lose that stability that we have associated with good health. We should also remember, of course, that we have multiple microbiomes. We just don't have one microbiome. We have different microbiomes in every region of the gut, for example, and we have different microbiomes in many different regions around the skin, and a different one in the lungs, and a different one in the urogenital tract. So we have many, many mini microbiomes inhabiting the human body. Generally, scientists look at the fecal microbiome because it's the easiest sample to get access to without doing biopsies or colonoscopies or very invasive techniques. But we should always remember that when we look at the fecal microbiome, we're only looking at one of our many microbiomes. And over the last 2 decades, the reason there are thousands of papers being published every year is that we believe now that the microbiome plays a very important role in health and in disease. So you may have as many as 2,000 different species in your gut, all unique to you. And these may play a role in a whole variety of diseases that I've listed here on the right. And this isn't an exhaustive list. There are many more than I've listed here. Now, this is not to claim that the microbiome causes all of these diseases, but all of these diseases are associated with significant changes in the microbiome, differences in the microbiome. The microbiome is a very complicated thing to study, and interpreting microbiome data can be very difficult. This is a paper we published a few years ago showing the microbiomes in elderly people, healthy elderly people living in the community, and elderly healthy people living in long-term care. And you can see the microbiome, well, you may not be able to see, but what this figure is meant to represent is the microbiome changes as elderly people move into long-term care. As their diet changes, as their health changes, their microbiome changes. And what we'd like to be able to do is to come up with interventions like probiotics, prebiotics, dietary interventions to slow down or prevent these health or these age and health associated changes in the microbiome. Here's another example in people with IBD or IBS, people with inflammatory diseases. This PCOA here on the left, again, these images can be quite difficult to interpret. I'll just bring up a pointer here for a second so I can show you. But these are people, these kind of red dots here they represent the microbiomes of people, control healthy people. And then as people get different inflammatory disease like IBS, ulcerative colitis, and Crohn's disease, you can see the microbiomes change in this direction. Again, we're not claiming that there's a single microbe driving these diseases, the very noisy signals, but there are obvious changes in people with inflammatory diseases. And once again, we wonder what would happen if we could drive the microbiota back in that direction. Would we restore health or at least reduce some of the consequences of these inflammatory diseases? We also learned relatively recently in the last decade or so that our microbiome plays a very significant role in mental health. This field has been championed to a large degree by two people, Ted Dinan and John Cryan, who are here in APC Microbiome Ireland, a psychiatrist and a neuroscientist. And what they and many others have been showing is that the microbiome plays an important role in stress and anxiety and cognition, learning behavior. So many of our brain functions are linked to our microbiomes as well. And of course, therefore, could be manipulated by probiotics and prebiotics and diet. So this opens up the possibility that the microbiome could be used as a therapeutic target. So if we accept that our health results from a combination of inputs from the host and inputs from the microbiome, then usually we try and direct interventions at the host. We use things like diet or lifestyle or pharmaceuticals to try and address the host. But what we're now realizing, of course, is there might be benefits, there will be benefits in addressing the microbiome. And there are lots more options we can use. In addition to diet and lifestyle and pharmaceuticals, there are lots of other interventions that could be targeted at the microbiome, including, of course, probiotics, prebiotics, postbiotics, fecal microbiota transplants, things of that nature. And so this has opened up a big opportunity in medicine to correct some of the chronic conditions of modern human life without resorting to pharmaceuticals and lifestyle and diet changes, or at least not relying solely on those. Of course, probiotics, postbiotics, things like that can also act directly through the host. We can reverse those arrows and say that if the microbiome plays a role in our health, then we can mine the microbiome for interventions that we could aim at human health. So we could, if probiotics can affect the microbiome and affect human health, then we could get probiotics from the microbiome that we could then use in other individuals. So the microbiome could also act as a rich source of therapies, could suggest smart diets, smart foods, smart feed ingredients like prebiotics that could influence human health. One example from a paper that was only published about a month ago by John Cryan and Ted Dinan and his colleagues showed that if you take feces from young mice and give them to very elderly mice, you can reverse some of the consequences of aging. You can reverse behavioral changes in aging. You can reverse immunological changes in the brain. So it gives us hope that we really can reverse some of the more dramatic effects of aging by using microbiome or microbiome-based interventions. And I encourage you to read that paper if you're interested in that area. But I want to focus largely on probiotics. Now, probiotics have been defined a number of times over the years. Possibly one of the more accepted ones is this one that we developed in 2014, which is just a minor change on an earlier one developed by the World Health Organization. And the definition is that they are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. And every word in that definition is important. They must be alive. They must be given in adequate numbers to have an effect. And they must have a demonstrated health benefit on the host. And I took this from another journal. There is high quality evidence, not just observational or people reporting feeling better, but truly high quality evidence for probiotics in a number of different gut associated conditions. And many of them are listed there. And the evidence, of course, varies across different conditions, but it is of good quality. So there's no doubt the probiotics really do work. But I should point out that they're tourists. They're not residents. Probiotics are not part of our microbiome. They arrive in the gut, they stay there for maybe a very short time, and then they are eliminated. So I use a comparison here of the Great Wall of China, maybe in a quiet time when there's very few people around, and then when tourists arrive, the wall is full of people or covered in people. The upper part of our gut, duodenum, jejunum, and ileum, contains relatively small numbers of bacteria, only about 10 to the 3 to 10 to the 7 bacteria. And we ingest probiotics in 10 to the 9, 10 to the 10 amounts. So temporarily after you eat a probiotic, the most abundant microorganism in your gut, in the upper reach of your gut at least, is the probiotic. So sometimes people ask, if we only eat probiotics at 10 to the 9 and our gut microbes are at 10 to the 14, how could they have any effect? But remember that the microbes are more sparse in the upper regions of the gut. So if we think about probiotics and microbiome, they're both living microbes, they share that. They both provide a health benefit, I think they share that. But they have a number of really significant differences. Probiotics are delivered to the body, whereas the microbiome is already present in all body sites. The probiotics are highly characterized, or at least they should be. For most probiotics, we have the full genome sequence of the strains. We know a lot about them, whereas the microbiome is extremely complex and difficult to study. And a very important difference is that probiotics are only present for a short time, whereas the microbiome is part of us essentially for life. It's in long-term residence. Once again, addressing this concept of, can these low levels of microbes impact human health? How can a small number of bacteria have an impact on such a big organism as a human? But we know already, of course, from medicine, from pathogens, that very low levels of microbes can cause significant health effects in humans, even lead to death. So we know that pathogens at very low levels can subvert the immune system, they can colonize, invade, produce toxins, make us very ill, and even kill us. So if we accept that small numbers of pathogens can cause us harm, it's not a big leap to accept that small numbers of probiotics can do us good. And we also wonder, of course, we understand a lot of pathogenic mechanisms. What are the probiotic mechanisms? And again, I'm making a case here that our health status results from an interaction between the host, our diet, and our microbiome. Those three things together give us our health. Probiotics can act through any of these three axes. They can act through the microbiome, make changes in the microbiome that influence host health. They can act directly on the host, of course, through the host immune system or enteric nervous system. And of course, they can act on the diet. They can enzymatically chew up parts of our diet or convert parts of our diet to other metabolites that have a health benefit. And once again, in the microbiome, we have a number of other options as well for affecting health status through the microbiome. But I'm focusing at the moment on probiotics. And something I want to really distinguish between is efficacy versus mechanism. Sometimes people complain for probiotics, well, we don't know the mechanism and therefore we can't trust them. But there are many drugs for which we don't know the mechanism. What we need to know to recommend a probiotic to a person with a disease or a person who is afraid of developing a disease, we need to know that they work, that they have efficacy. As scientists, we'd like to understand mechanism because we like to understand things and we'd like to select even better probiotics. But efficacy, evidence of efficacy must be generated in a targeted host, in a human, for example, in an appropriately powered trial. So you must confirm efficacy and you must demonstrate that to the regulatory authorities' satisfaction in order to make a claim. And of course, the evidence must be in the target condition and at the correct dose. You can't show a probiotic works in treating diarrhea and then recommend it for treating anxiety. You have to show that it works in both conditions before you recommend it for both. But mechanisms are also important. And as scientists, we always want to understand mechanism. And these are usually done in model systems, like rats or mice or cell line systems. Very often involves knocking out genes or introducing genes into probiotics. These are intellectually satisfying. They support regulatory applications. They help to select which targets we should apply a probiotic for. But as again, they're not necessary to prove that a probiotic works. But we love to know mechanisms. But sometimes, again, people criticize papers about probiotics and say, oh, that was only done in mice. But if it was done to establish mechanism, it's entirely appropriate it was done in mice. If it's done to establish efficacy, then it really should be done in humans. There are some misconceptions about probiotics. Probiotics do not have to colonize the host. There was a major paper published in Cell in 2018, which said, and it was reported as this, human gut study questions probiotic health benefits. They may not be as effective as we think. And what the scientists did was they said, surprisingly, we saw that many healthy volunteers were resistant in that the probiotics could not colonize their GI tracts. And this suggests that probiotics should not be universally given. Well, I'd agree with the second part, but the first part is a mistake. Probiotics do not have to colonize the host. There's nothing that says in the definition that probiotics have to colonize. They do not have to grow or demonstrate metabolic activity within the host. They may interact directly with the immune system. They do not have to impact on microbiome composition, and they do not have to have a known mechanism of action. We would love to know all of these things, but we don't need to know them in order to claim that something is truly a probiotic. So probiotics can be complicated. There may be some general benefits that are widespread among studied probiotics. It may be that most probiotics have a certain beneficial effect, and these beneficial effects may be kind of broad and general. There may be some benefits which are more specific, and maybe only some probiotics have those effects. And there may be yet other benefits that are very rare, and only one probiotic or a very small number of probiotics might have that benefit. And so it's not a conflict to say that some probiotics might work in many people and some probiotics may only work in very specific people. It will depend on the probiotic. So here's an example of widespread efficacy. This is a meta-analysis that was done quite a while ago now, almost 10 years ago, by Ritchie and Romanuk. And what they looked at was the efficacy of probiotics across a huge range of conditions. Every one of these lines here is a clinical trial. If it's to the left of this line, it means it was beneficial. If it's to the right of this line, but doesn't touch the line, then it means it was detrimental. And what they showed was, and it's written here, 74 studies, 84 trials, 10,000 patients showed that in general, probiotics are beneficial in the treatment and prevention of GI diseases. Across a whole range of probiotics, there were generally beneficial effects. So that's widespread benefits associated with probiotics. There are some benefits that are more frequent but not widespread. So for example, in acute diarrhea, here's a meta-analysis that shows that, for example, some probiotics have essentially no effect. Some probiotics are quite good. Other probiotics have no effect. So it's not this broad benefit, but there is quite frequent benefit for this particular condition. And then there are some conditions where you really only have strain-specific efficacy. This is the ESPGHAN working group on prebiotics and probiotics. And what they say is that for nosocomial diarrhea, for hospital-acquired diarrhea in children, only one probiotic seems to work to a level that they would support its use in medicine. So complicated, but some probiotics may give very general benefits. There may be some probiotics which give kind of frequent benefits, and then there may be some benefits that are only given by the right probiotic for the right condition. And this is emphasized by a nice paper from Michiel Claerbesem's lab, where he looked at, this is not something that people commonly do, but he went in and took biopsies of people's gut after they had received a probiotic. And what he could see by looking at the immune responses, looking at things like cytokine readouts, he could see that the same individuals took all 3 probiotics at different times, reacted very differently to the 3 probiotics. So to Lactobacillus acidophilus, Lactobacillus casei, and Lactobacillus rhamnosus, there were 3 completely different immunological responses to those probiotics. So we do detect and respond to probiotics selectively and specifically. Not all strains are the same. And this is emphasized by lots of groups, but I'm just pointing out here the World Gastroenterology Association or organization guidelines issued a few years ago. This is a table that I'm showing here, but the table goes on and on and on. And what they're taking is different conditions and listing the probiotics that have been shown to have a benefit in those conditions. And also giving them their evidence level, their level of faith in these. So 1 is the highest level of evidence, 2, 3, 4, down to 5, and so on. So it's a very useful set of tables if you're interested in finding which probiotics are associated with which conditions or which are likely to work in which conditions. But as scientists, we're always interested in mechanisms. And people have speculated for a long time what kind of mechanisms might be involved. And it could be, of course, that probiotics may produce different kind of effector molecules. Life, in the end, is all about molecules interacting with molecules. And so probiotics must produce molecules which interact with human molecules to give a health benefit. Some of these molecules may be delivered in the initial dose, things like bacterial structures or bacterial biomass. Some may require active metabolism in the GI tract, such as, for example, bacterial metabolites. The bacterium may have to grow, the probiotic may have to grow in the gut in order to give these benefits. We have been able to show, this is a paper by Joseph et al., which showed that probiotics can, in addition to influencing our immune system, as Michiel showed, can also influence our microbiomes. So here you're seeing children be given Lactobacillus casei shirota, which is a well-known probiotic, and seeing increases in other Lactobacillus species and in Bifidobacterium over time in response to getting a Lactobacillus casei. So there's an influence on our microbiome by the ingestion of these probiotics. Now, this particular intervention here was not linked to a health benefit, but Bifidobacteria are generally regarded as being beneficial bacteria in the gut. Bacteria can produce enzymes that are probiotic factors. For example, in the disease of lactose maldigestion, a lot of people on earth cannot digest lactose as adults, and they suffer if they drink milk. You can take bacterial enzymes that will resolve those, or you can actually just take yogurt cultures, which produce a lot of the enzyme lactase, and that will help you to digest the lactose and relieve those symptoms. So this is the only probiotic claim that has been approved by the European Food Safety Authority, and we know the mechanism which helped to get this claim approved. I'm very interested in probiotics and infection. How could probiotics help to prevent against infection? And it could be, of course, that probiotics could directly kill the pathogen. They could work by direct antagonism. They may work by immunomodulation, as I've already shown. The probiotics could interact with the immune system in such a way that it makes the immune system more resistant to infection. Or they could work through competitive exclusion. They could influence the microbiome or tight junctions or mucus production in such a way that the pathogen is excluded. All these mechanisms are possible. Now, we looked in a particular model system for a mechanism for this particular, for an anti-listerial mechanism. So listeria monocytogenes is a very potent infectious bacterium. It infects humans and causes fatalities in about 20% of the humans it infects, and it also infects mice. So what we did was we infected 6 groups of mice, I'm just showing one mouse from each group, with a Lux-tagged Listeria monocytogenes. The monocytogenes was genetically modified so that it emits light. And you can see the Listeria through the body of the living animal. And you can see that the liver is becoming infected, and eventually the brain will become infected and these mice will die. Now, all of these mice were given different, or the 6 groups of mice were given 6 different probiotics. And 5 of those probiotics had no effect on the Listeria infection. But one probiotic completely prevented infection. That was Lactobacillus salivarius UCC118. So this is showing efficacy only in a mouse model, but efficacy nonetheless. When we looked for mechanism, what we saw was that this particular bacterium, UCC118, can produce a bacteriocin. This is a small peptide that can kill Listeria monocytogenes. And this was an obvious candidate to be, to be the mechanism by which UCC118 was killing the Listeria. So we created a genetic knockout which could no longer produce the bacteriocin. And then we tested that in the same mouse model. And what we saw was that when there was no probiotic, the mice were significantly infected with Listeria. When the probiotic was present, we got no infection. But when the probiotic that didn't produce the bacteriocin was present, full levels of infection were restored. So in other words, we can say that it's the production of this bacteriocin is the mechanism by which UCC118 protects these animals. And it's a rare instance where a single molecule has been identified as a probiotic mechanism. There's another example from our own lab, our colleagues' lab here in APC Microbiome Ireland. This is a bifidobacterium, again, that has been engineered so that it's emitting GFP, and you can see the beautiful shape of the bifidobacterium. And what they were able to show was that this particular bacterium has a very important effect in wound healing in the gut. They also showed that it produces a pilus. This bacterium produces a long pilus, and that pilus contains a particular protein called TAD-A. If they knock out TAD-A, the knockout no longer has the probiotic effect, just as we showed by knocking out the bacteriocin gene. And purified TAD-E, which is the protein produced by the gene TAD-A, can recapitulate the effect, has the same effect as the original probiotic. So the original probiotic has a wound healing effect that's lost in the knockout, but you can get the same wound healing effect from that single protein. So once again, a single protein responsible for a probiotic effect. And it's very satisfying when we can bring it down to a single protein, because now, of course, we can look for other probiotics that also produce this protein. We're entering a very interesting phase of probiotic research too. We're looking at next generation probiotics. So traditionally, probiotics were things like bifidobacteria, lactobacilli, even yeast, and even E. coli. But now, of course, we're starting to mine next generation probiotics from the microbiome. Bacteria that we had no history of using before, like Akkermansia, Bacteroides, Faecalibacterium. We're even looking at genetically modified microbes as next generation probiotics. So it's a very exciting time for probiotic science. And here's one example of a next generation probiotic, Akkermansia mucinifila. This has been shown to have an effect in mice, in overweight and obese mice, in reducing metabolic syndrome in those animals. And it has now moved on to IND. So I'm getting some feedback from someone's mic. It's moved on to investigation of drug status with the CDC, and it's going into randomized clinical trials. And it has also shown effects in humans. So I'm coming to an end now. So what I want to say was that ISAP, the International Scientific Association for Pre and Probiotics, has done a lot of work on trying to define these different terms like probiotics and prebiotics and postbiotics. And I'd refer you to these papers if you're interested in any of those definitions. The definitions are hopefully pretty clear, but you should really read the whole paper to understand the complete concept. So prebiotics that I've mentioned up to now, they're substrates that are selectively used by host microorganisms conferring a health benefit. So prebiotics are aimed directly at the microbiome to try and promote the growth of beneficial bacteria in the microbiome and give a health benefit. We also have symbiotics. Symbiotics are a mixture of live microorganisms and substrates selectively utilized by host microorganisms. So essentially probiotics and prebiotics that confer a health benefit. And there was a lovely demonstration of that by Pinaki Pinagrahi's group in India, where they showed that delivering a symbiotic, Lactobacillus plantarum and fructooligosaccharides, in rural India to prevent sepsis among infants. This is a very large trial of 4,500 infants. They observed a very significant reduction in death from sepsis in those infants. Postbiotics, I just want to spend a couple of seconds on postbiotics. Postbiotics are inanimate microorganisms. So they're bacteria that have been inactivated. And these are safer, perhaps, than living organisms. They may be more easy to dose. They certainly be more stable. And of course, they can't be cultured from the product. So that could have intellectual property considerations. Postbiotics can be effective as well. For example, Akkermansia mucinifera is a probiotic. I showed you that already. But it's also a postbiotic, because what they showed in this paper in Nature Medicine was that if you inactivate the probiotic by pasteurization, so you have a heat-killed probiotic, that works not only as well, in fact, it works better than the live bacterium. So the dead bacterium has a better beneficial effect than the live bacterium. So this is a postbiotic. And they were also able to go on and show that it was actually a single protein on the surface of those bacteria that could at least partly recapitulate the beneficial effects. So mechanism, but in this case, it's also a postbiotic. And there are many other examples of postbiotics. I won't go through them, but for a lot of different conditions, there is evidence that dead bacteria can work. And that shouldn't really surprise us, because there's, of course, a lot of molecules already present in these dead bacteria that can interact with our microbiomes, with our immune systems, with our nervous systems to deliver a health benefit. Dead bacteria can also cause disease in the case of intoxicative organisms. So we're well used to the idea that already produced metabolites or structures can have a health effect. So in conclusion, I think we're in a really good place. We have a virtuous cycle. We as scientists want to do the highest quality science. And of course, we want to make an impact on society. We want to change things for the better. Our industry partners want quality products with validated and understandable claims. And our regulators, who are sometimes presented as our enemies, are not our enemies, of course, they're our partners. They want to protect consumers from misinformation and reward scientists and industry who do the best quality work. And our clinicians and our dietitians want to reliable information to make informed decision to inform consumers. And of course, consumers want to be able to trust the claims that they see on products or the things that their clinicians tell them. So it's a perfect circle. If everyone does high quality work, then everything works out in the end and delivers the right thing. And just to emphasize that, Akkermansia mucinifera, which I just talked about, has actually now, just a matter of months ago in July 2021, is the first next generation bacterium to be given status in the EU. Now, it's just as a novel food, and it's only been designated as being safe, not as being efficacious yet, but it is a real breakthrough that the regulatory authorities, because the work was of very high quality, are accepting that these probiotics and next generation probiotics and postbiotics have a role to play in medicine. So my first conclusion is that probiotics and postbiotics and symbiotics can influence human health either directly through the immune system, the nervous system, by having enzymatic activity, by producing metabolites. And prebiotics, and again, postbiotics and probiotics and symbiotics can also influence human health indirectly via the microbiome. So we have lots of options. Our end goal, of course, is always to improve health, but we're opening up lots of new options of how we can do that through the development of prebiotics and probiotics and postbiotics. And so my second conclusion is that probiotics are an evidence-based alternative, not an alternative medicine. But of course, we must choose the most appropriate probiotic for each specific condition. There may be general benefits from the consumption of high levels of probiotics across a number of conditions, but if you have a condition, seek out the best probiotic for that condition. So I want to thank my colleague, Professor Paul Ross, who you see in this picture here, who is my collaborator in nearly all of the work that I've talked about. Of course, my two parent institutes, APC Microbiome School of Microbiology, and you, of course, for your attention, and you for the invitation, which, as I said, was a tremendous honor for me. Thank you very much, and I'd be glad to answer any questions you might have.
Thank you, Professor Colin, for such an enlightening and entertaining talk on the role of probiotics as an alternative medicine. The knowledge that you have shared has helped us immensely in improving our understanding of probiotics. Well, the first question that I have for you is, there have been a number of studies related to the gut-brain axis that would aid in understanding and treating neuropsychiatric disorders, especially depression. Do you think we can safely draw the conclusion that since a poor diet is a risk factor for depression, a healthy diet can prevent depression? The reason I ask this is the emerging role of psychomicrobiotics for the treatment of psychiatric disorders. Yeah.
Yes, I mean, we always make the mistake, I suppose, when we're talking about probiotics, we focus on probiotics and seem to give the impression that probiotics are the only answer. And of course, they're not the only answer. Diet is hugely important in many, many different diseases. And diet shapes our microbiome. Diet obviously informs our nutritional status. And probiotics have to work against the background of diet and the microbiome. And so I think a combined approach, as we just suggested, I'm not sure that probiotics have been really shown to work in severe depression, in real depressive disease, but certainly in cases of anxiety and maybe pre-depression kind of symptoms, there has been a significant benefit for both diet and probiotics. But I do think, you have to treat people holistically. If someone is depressed, you can't just flick a switch with a probiotic and cure them. You have to really address all of the problems surrounding that depression, which would include environment and diet and perhaps microbiome-based therapies as well.
So maybe doctors will start prescribing probiotics instead of sertraline for depression in the future, along with the
anxiety. Anyway, there is a product on the market here in Ireland, at least, and in Europe, for treating again, anxiety and stress, not depression, but anxiety and stress. A probiotic, lactobacillus, that produces GABA, which is a neurotransmitter which is known to be involved in, obviously, the transmitting of signals between the gut and the brain.
Right. Professor Colin, we understand that every probiotic produces multiple molecules which can interact with the microbiome and the host at multiple points, and not a single molecule mediating the effect on the health of the host. So it's not as if we can tinker with one molecule to modify the probiotic effect. Do you think this complexity and also the fact that we work with animal models and not human models, this is responsible for limiting the use of probiotics as potential one-stop treatment for diseases the way traditional drugs are being used?
It's a very good point. Probiotics are essentially almost polypharmacy because they're producing many different molecules which interact with the host. I do think for some health indications, it might all come down to one particular molecule being presented in a particular way on the surface of a probiotic, for example. But for many of the effects, they must be multifactorial, lots of different interactions. But that's not necessarily unusual. I think even for small molecule pharmaceuticals, they don't only have an on-target effect, they're very often off-target effects as well. And we just have to manage those and make sure that the benefits accruing from the positive effects outweigh the negative benefits or any off-target effects, if you like. I don't think probiotics are likely to be a one-stop shop for any complex medical condition. But I do think they can help in maybe maintaining remission after a drug treatment, for example. Maybe you need drugs to get you into remission, and then probiotics could keep you in remission. They may prevent the condition from developing in the first place, and they may relieve symptoms associated with a particular condition. But obviously they can never reverse a genetic disease, or they can't reverse a disease which has many, many causes.
As you said, different probiotics work differently for different people. So do you think it is possible in the future to have personalized therapeutic recommendations using a sequencer that you can directly connect to your phone, and then that could give you all the information regarding what type of microbiome you have, what infection you have, and maybe the best possible treatment for it, or the best possible probiotic?
It's a wonderful dream, and we should dream big. I mean, we have a sequencer in our lab, I'm sure some of you have in your lab, where we can put a sample into the sequencer, connect it to our mobile phones, to an Apple phone, and literally watch the sequences be just read off in real time. So I think we are approaching a time where we will routinely sequence someone's microbiome as part of our triage, if you like, to decide which chemicals you would use. I mean, it's been shown, for example, that the checkpoint inhibitors, which are a wonder anti-cancer drug, don't work in some people entirely because their microbiome prevents them from working. And so knowing somebody's microbiome will be a very important thing for choosing the right medicines and choosing the right probiotics. You could maybe look at someone's microbiome and see that they're deficient in a particular enzyme activity, and we could give them a probiotic that delivers on that enzyme activity, like lactose intolerance, for example. So I think it's a dream, but I think it's a closer dream than we might think. I think, in 10 years' time, 20 years' time, doctors will say, can you imagine there was a time when we never paid any attention to the microbiome and we never thought about using the microbiome as an intervention? It'll seem extraordinary, I think, in the future.
Truly. One last question. Is there any concern that the next generation probiotics and live biotherapeutic products, they might face the same fate in terms of consumer perception as the highly controversial genetically modified food did? And should we start educating the children and the other households about the use of the probiotics at the earliest?
Yes, I think you're absolutely right. I mean, I think people are afraid of the unknown anyway. And so while they seem to trust pharmaceuticals, to a degree and just accept them without questioning, bacteria have been given a very bad name in society. So we see ads on our television, at least, proudly saying that something will kill 99.9% of all bacteria. And we associate bacteria with unhygienic conditions and dirt and not being clean. And so people already have a kind of a bad impression of what microbes are and what they do. And so it'll take some work to convince people, and maybe like the first slide I gave, persuading people that we live in a microbial world, and without our microbes, we would have nothing. We'd have no oxygen to breathe, we'd have no food to eat. Microbes shape our world and they shape us, and we need to harness them and make them our friends.
Thank you, Professor Colin. Just one last question. There are a number of questions from our listeners. Probiotics have a short-term exposure. This short-term exposure is a result of probiotics attaining stationary phase, or is it because that our local gut microbes compete with probiotics, or is there any other reason to that?
There may be any number of reasons. I think it's probably the second. I think it's probably competition. I mean, when you think about it, a probiotic has been probably grown in a stainless steel vessel, and then it's been dried and then packaged in a capsule or put into a food, and then it arrives in the gut. It's not adapted to grow in the gut. It hasn't grown in the gut for a very long time, if you like, for many generations. It's probably not expressing the right proteins on its surface. It's not adapted, and it arrives into an environment with many bacteria that are already adapted, and I think it just simply can't compete. Some of them can metabolize, but very few compete. It does happen. Some people do get colonized, but it's quite rare. So I think it's competition.
Thank you, Professor Colin. Thank you for answering all our queries. Now I would request Dr. Archana Vora to deliver the vote of thanks. Dr. Archana Vora, thank you.
Yeah. Thank you so much, Professor Colin, for sparing your precious time and sharing your research expertise, your knowledge with our students and with the faculty members. I also extend my heartfelt thanks to our director, Dr. Geeta Trilok Kumar, for always encouraging us in organizing more such webinars, and the entire team behind organizing this webinar, Professor Deshmukh, Dr. Archana, Dr. Sunita Agarwal. With their enthusiasm and their efforts, they have very successfully organized this webinar. And thanks to all the faculty members and the participants. Thank you so much.